Hello everyone, welcome to today's live broadcast, Automated Single-Cell Dissociation of Primary Tumors and Patient-Derived Xenografts. I am Judy O'Rourke of LabRoots and I'll be your moderator for today's event. Today's webcast is presented by labroots.com, the leading social media site for science professionals, and sponsored by Miltenyi Biotech. Miltenyi Biotech is a global provider of products and services that advance biomedical research and cellular therapy. The company's innovative tools support research at every level, from basic research to translational research to clinical application. This integrated portfolio enables scientists and clinicians to obtain, analyze, and utilize the cell. Miltenyi Biotech's technologies cover techniques of sample preparation, cell isolation, cell sorting, flow cytometry, cell culture, molecular analysis, and preclinical imaging. Their more than 25 years of expertise span research areas including immunology, stem cell biology, neuroscience, and cancer, and clinical research areas like hematology, graft engineering, and apheresis. The company also offers comprehensive scientific support, consultation, and expert training. To learn more about Miltenyi Biotech, visit www.miltenyibiotech.com. Before we start, there are a few instructions. We want to hear from you during this interactive broadcast, so please ask questions or leave us a comment. Answers are welcome too. You can do this by hitting the green Q&A button at the lower left of the presentation window and typing in your comments and questions. We'll try to get to as many as we can, and we'll follow up if we don't have time today. Want a better look? You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you can't hear or see this presentation properly, let us know by clicking on the support button on the top right or use the Q&A button. We'll make sure we try to resolve any issues. Now let's get right to today's presenter. We are proud to welcome J.T. Poirier, Ph.D., an assistant laboratory member at Memorial Sloan Kettering Cancer Center Medicine. Dr. Poirier's lab focuses on the development of new patient-derived xenograft models and experimental lung cancer therapeutics. During his career, he has developed a wide range of skills in laboratory-based and computational research. Having these hybrid skill sets positions him to operate at the interface between the world of the wet lab and that of the high-performance computing cluster. Dr. Poirier's research has focused on the genetic and epigenetic landscapes of small cell lung cancer, therapeutic targeting of the DLL3, and development of oncolytic viral therapies. He has also established and oversees an active program in patient-derived xenograft development and characterization. Through this program, he has developed a series of best practices for genetic characterization of human xenografts grown in mice and biomarker validation in PDX. I will now turn the presentation over to Dr. Poirier.
Thank you. This presentation is going to cover one specific aspect of our research program that we're finding that we use fairly often, and that's automated single-cell dissociation of primary tumors and patient-derived xenografts. There are two learning objectives for this presentation. One is I want to cover the advantages and challenges associated with using PDX models for cancer research, as well as solid tumor dissociation methods compatible for downstream single-cell applications such as flow cytometry, immunomagnetic enrichment, and single-cell sorting. So just a one-slide introduction about my lab and my research interests. My interests really focus on the lung and lung cancer. Lung cancer is an extremely lethal and devastating disease, and it's a disease that we focus our research on, starting with a important clinical question, then taking this to the basic science laboratory, performing validation studies, and then hopefully bringing this full circle back to the clinic. And in the basic science applications, as you've heard, my lab does a lot of genomics, epigenomics, and transcriptomics. We're recently doing more functional genomics using genome-scale CRISPR, and we've traditionally done a lot of basic biochemistry and molecular biology. In the validation phase, I'm very interested in high-fidelity cancer models, particularly in mice, basic pharmacology, and more recently molecular imaging. And then on the clinical side, I partner with clinical collaborators for early phase clinical trials, retrospective biomarker studies, and diagnostic assay development. This talk is divided into a few different parts. I'm going to start off by just giving a short introduction to patient-derived xenografts. We think about xenografts in mice as taking a couple of different general approaches. The standard xenograft model for several decades has been to take primary tissue from a patient from the clinic, establish a new immortalized cell line grown in cell culture, and then graft those cells into immunocompromised mice. By contrast, patient-derived xenografts never are grown in vitro, and one of the major differences here is that there's a selection process when bringing cells from the in vivo environment to the in vitro environment that is irreversible when you bring it back to the mouse. And we think that this is driven by changes in oxygen tension, growth substrate, growth factor, and nutrient gradients, among other things. So my lab has been really focused on using patient-derived xenografts as a tool for concept validation in a preclinical setting, and using that information in a push-pull mechanism with the clinic. So there are a number of different mouse models for cancer, and they all have we think they're really complementary, and each one has their own advantages and disadvantages. I told you about traditional cell line xenografts. These are human cell lines. They're relatively fast and cheap to work with once the cell line is established, and they're fairly easy to engineer with lentiviruses and other similar technologies. Patient-derived xenografts are similarly human. They're relatively easy and cheap to work with. We think they maintain a slightly better, more high-fidelity model of primary tumors, but they're generally much more difficult to work with than cell line xenografts. I won't be talking about these today, but I'll just add that, of course, there are also genetically engineered mouse models, or GEMs. They have distinct advantages in being used in immune-competent settings, having more heterogeneity in the cells that are initiated. They're much more facile engineering of the genomes in these models. And of course, you can take cells from these GEM models and engraft them directly into syngeneic mouse, which is sometimes called a gem-derived allograft, or a GDA. This presentation is really going to focus on PDX, but some aspects I think will also be applicable to traditional cell line xenografts in mice. I just want to illustrate on this slide that when we think about tumors, they're really a composition of cells that include the tumor cells themselves, but also various cells of the immune system that will infiltrate the tumor, the vascular network that brings nutrients and oxygen to the tumor, associated fibroblasts and other cells, and that the tumor mass itself is made up of human cells that are the cancer cells themselves, but all of these support stromal cells come from the host, which in the experimental setting is mouse. And this imposes several challenges. One is that it can be difficult to control the tumor growth variance within treatment groups during the therapeutic studies. You can have some tumors that grow faster or slower. Another problem with having admixed mouse and human cells is that the nucleic acids that you extract from a bulk tumor are actually both human and mouse origin, and I'll show you that the murine component can actually make up a very significant fraction of the nucleic acids that you extract. The other aspect that can be challenging is that manual disaggregation may not be technically consistent for single-cell sequencing applications. And I'll show some QC data of some of our initial experiences with single-cell RNA-seq. When we first started working with PDX, our general approach was we would put some tumor tissue into a petri dish like this in a small amount of media, and we would take a razor blade and just chop up the tumor into as fine pieces as we could, put this through a nylon mesh strainer, and then hopefully that would dissociate the tumor to a point where we could inject this into mice to establish a cohort. And over the last few years, when I established my lab at Sloan Kettering, we've been using an automated method to try to make this process more consistent. And to illustrate the instrument that we use for dissociation, which is a Milton E. Octamax gentle dissociator, I have a 15-second video queued up if you could play it, please. Basically put the tumor into these dissociable tubes. We add an enzyme mix, and the tissues then get disaggregated in a programmable and reliable way. And I'll just show a couple of quick slides about the effects that this can have in animal experiments. So, a few years ago, before we started using this approach, it was sometimes difficult to control variability within treatment groups. So I'm showing here tumor growth curves from an in vivo xenograft experiment using the same PDX line. And on the left, we had really very little variability within individual groups, and this made it really, after only a few days of treatment with an experimental therapy, to see that there's a significant difference in growth between these two groups. But the same experiment repeated had a much higher variability and was very much more difficult to interpret. And we wanted to have some better control over this by dissociating the tumors in a reliable way so that we're not having some mice that get more or less tumor tissue than others. This is a in vivo experiment from a recent publication where we used automated dissociation. I think you can appreciate that in this very large experiment, the variability within groups is much, much smaller. The other approach that the other application that we're finding for this dissociation is in quantitating the amount of mouse stromal cell contamination that there is, and actually doing some testing approaches to remove those contaminating cells. So I told you that the tumor xenograft is made up of a varying, a human cell component and a mouse cell component. And so what we wanted to do is be able to take a xenograft from a mouse, perform automated dissociation, and then study these different components independently. So one of the first things that we did was just ask the question, in our lung patient-derived xenografts, how much, what percentage of the tumor was actually made up of mouse cells versus human cells? And on the far left, you can see a flow cytometry experiment we did where we stained the cells, we double stained the cells either with an epithelial marker, in this case EpCAM, or a mouse-specific marker, H2KD. And you can see that once you get a single-cell dissociated population, you can really separate the cell types into two really distinct groups. And this let us do a couple of things. One is it let us enumerate the number of cells that belong to each class, but it also gave us access to the potential to use flow cytometry to sort these two populations and obtain pure reference populations of either one. We've also been using a technique that was developed by Jim Eshelman and Sarah Whelan, who are former colleagues at Johns Hopkins, that uses a PCR amplicon-based approach to actually quantitate from a mixed pool of nucleic acids how many or what percentage of those nucleic acids came from a mouse genome versus a human genome. And in the middle here is just shown two different peaks. The blue peak is from the human cells, and the orange peak results from a PCR product from the mouse cells. And so we were able to, by quantitating the peak height, figure out how much contamination there was. And it was gratifying to see that once we started to calculate the amount of contamination, that in the lung adenocarcinoma PDX, we saw that the tumors were more diffuse and more infiltrated with stroma than, for example, in small cell lung cancer. And this really reflects what the histology of these tumors looks like. And part of why we started to think very hard about this problem of contaminating mouse cells was that we were starting to, it was our endeavor to try to have as careful molecular characterization of these xenografts as we could, and we wanted to implement a targeted sequencing assay that's now used in the clinic at our institution called MSK Impact to characterize these samples. And I won't go over all the details of this assay because it's published, but the concept is that biotinylated probes allow for hybridization in solution with targeted exons from a small number of cancer genes. I think it's now more than 500 or 400 cancer genes. These are sequenced to very high depth to allow for high sensitivity, and then a mutation calling pipeline is able to call mutations. So this is an example of a tumor that's sequenced compared to its normal, and we're looking at just one exon from the EGFR gene that harbors the activating L858R mutation, and this is showing the sensitivity of this approach to detect this, this activating mutation in cancer cells. I won't go over this, but this is just a slide showing some of the major cancer genes that are covered by this panel. And I'll also just note that this panel can be used to call copy number as well. So this was really attractive to us, and we wanted to be able to apply this sequencing approach and other sequencing approaches like whole exome sequencing to our growing library of patient-derived xenografts. And one thing that all of these technologies rely on is alignment of nucleic acids and short reads to a reference genome. So I already told you that these tumors are composed of nucleic acids from both the human cells, but also from the mouse genome. And what I'm showing here is just a cartoon that describes the problem of trying to sequence a mixed population of two different species. So if you envision aligning reads from a purely human sample to either the HG19 human genome or the MM10 mouse genome, I think it's intuitive to think that the majority of these reads are going to align very well to the human genome. But we also know that there are conserved sequences between human and mouse, and some human reads may actually align to the mouse genome, which is shown with the blue reads overlapping the orange. But I think what happens when you replace a portion of those human reads with some contaminating mouse reads is you have the converse. So some of those reads never align to the human genome, and some of those reads will align better to the human genome than they do to the mouse. And this results in really two complications. One, I think you can appreciate, is the loss of total depth of reads that align to the human genome. But also, this is a problem to have mouse reads that are partially mapping to the human genome. And I'll illustrate exactly the problem that this causes in real-world samples later, but for now, take my word for it that this can cause a very high rate of false positive mutation calls. So you want to get rid of these reads, and we took two approaches to this. And one traditional approach had been to do a two-pass mapping approach, where you map all of your reads to the human genome, then you map all of your reads to the mouse genome. You set cutoffs for identity, and then you eliminate the reads that map better to human or mouse than to human. We've been taking a different approach that we call a hybrid genome approach that does a single pass mapping against a hybrid genome that includes all human and all mouse sequences. And then after the mapping is done, we just discard reads that map better to the mouse or preferentially map to the mouse genome. And we wanted to benchmark this approach and using as real-world a sample as we could. And so we turned to the Illumina Platinum genomes. And these are a trio of individuals that has been sequenced to very high depth on an Illumina HiSeq 2000 system. So these are 200x depth. For one of the samples, it was there was a technical replicate, so it was sequenced to 200x depth twice. And because this is a trio, there's very high confidence SNPs being able to call, very high confidence SNPs called for one of the samples, with phasing from the two parents. So we started by downloading these genomes, and what we did was align them either to the human genome, which is on the x-axis, or to the hybrid genome, which is human and mouse. And then we examined what the read depth was at every base. And what we were concerned about is what's highlighted in this triangle, which is basically we wanted to estimate the potential loss of depth at different locations in the genome, because we didn't want regions of the genome to go dark because of too high homology between human and mouse. And our conclusion from this was that while there's some loss of depth in certain loci, it's not the loss of depth is not so great as to prevent accurate mutation calling, and we can still have greater than 98% of the bases in the genome are callable, which is about the same fraction whether you're looking at a hybrid genome or looking at the human reference alone. So these in silico experiments kind of convinced us that this could be a usable approach, but we wanted to really get at what the operational effect of mouse contamination was on genome sequencing pipelines. So we turned to this great tool that was published in Nature Methods in 2015 called BAMSurgeon. And this tool was created as a way to generate gold standard reference files with mutations in known locations. And this was used as a way to basically benchmark different mutation calling algorithms in the ICGC mutation caller shootout. And what we did, the advantage of this is basically it takes an already aligned BAM file. It also takes a number of genomic loci where you want to insert a mutation, and it inserts those mutations and then remaps the reads that contain the mutation. So what you end up with is a BAM file that has been altered to have mutations in known locations. And so it allows you to then go back and ask if your mutation caller is able to recover those mutations, and to what extent your mutation caller calls mutations that don't exist. And so we came up with this pipeline that we internally called the Mouser, and this diagram is a little bit complicated, but what is really being shown here from the top is that we start with a list of single nucleotide variations that we were interested in studying. And so these were a list of known mutations at known loci that we wanted to spike in. And then we started with the technical replicates of one of the Illumina Platinum genomes at 200x depth, and a 40x genome from the NSG mouse. And using these three sequence genomes, what we did is basically feed the single nucleotide variants and one of the technical replicates into BAMSurgeon to insert mutations, and then use what we call the Mouser to randomly mix in a variable amount of mouse genome sequences. So what we end up with is, on this row, what we call a virtual tumor at 60x, a virtual PDX also at 60x with some amount of contaminating mouse reads, and then a normal at 30x. So all we did here was downsample. And basically we ran these into Mutect, called single nucleotide variants with Mutect, and then we used a comparator algorithm to compare the mutations that we put in there artificially and the mutations that we were able to get out in the end. And what this pipeline ended up spitting out for us was this figure, which is a small figure, but it says a lot of things. On the x-axis is basically the amount of mouse DNA contamination increasing from left to right. On the y-axis is the F1, which is the harmonic mean between sensitivity and specificity. So the range here is from 0 to 1, and 1 means that you have both high sensitivity and specificity. The lines here are showing mutations that are inserted at different varying allele frequencies. So the blue line, we're looking at a mutation that is spiked in at roughly 5% varying allele frequency versus around 40% varying allele frequency in orange. The open symbols here are looking at exome or exonic territory, whereas the closed symbols are looking at the same variant frequency in whole genome territory. So I think we took a couple of things away from this. One was that at 30x to 60x depth, 5% varying allele frequency, you weren't really going to be able to reliably call those mutations anyway. At high varying allele frequency, you can tolerate a lot of mouse contamination, but the low frequency variants in the 10 to 20% range, the ability to call those mutations really drops off as your mouse contamination increases. And we think that this is really driven by loss of overall total depth. So as your sequencing depth drops, you really lose the sensitivity and specificity. So to mitigate this, we realized the only thing that we could really do here was to get rid of the mouse stroma. And so we decided to audition different techniques to be able to remove these cells. So what we did was dissociate some tumors, and then audition different approaches to purify out the tumor cells, and then use as our gold standard quantitation by a PCR amplicon-based approach. So what we did for 5 different PDX was compare the pre-sort, which is the input, to fax sorting, which we considered to be our gold standard. Then we looked at a negative depletion method, which pulls out the mouse cell, a mouse cell depletion kit, which pulls out the mouse stroma, or we looked at positive selection approaches, which would pull out the tumor cells of interest. And our conclusion based on this was that fax is a great way to purify your cells, but we really like this mouse cell depletion approach. We think it's much more powerful for removing cells. And I think that our conclusion, especially looking at MSK-LX27, was that if you're looking at a model that initially has fairly high mouse contamination from the start, it's really hard to, using positive selection, pull out a very pure population. But with a negative selection approach, like mouse cell depletion or fax, we were able to get relatively pure populations. And so I said that I would go back to the question of real-world example of using these approaches. And so we sequenced a couple of patient-derived xenografts, either comparing the pre-sort input variant allele frequency to the variant allele frequencies of different mutations called in a sample that was purified by fax, and that's what's shown on the left here. And I think you can see the dots that have open circles here fall on the unity line, which means that those are mutations that are called in both samples, and they're called with relatively similar variant allele frequencies. This is in contrast to private mutations that are called only in the input that are shown in red. And so they have essentially zero variant allele frequency in the fax, which means they're not detected, but they're detected in the input. And these represent 187 false positive mutations in this one sample compared to the 9 true positive. And these false positive apparent mutations are really just mouse reads that are mapping partially to the human reference genome. This is in contrast to if you use a mouse cell depletion kit and compare this to fax, you really see that they perform equally well at removing these mouse cells and preventing this high false positive detection rate. And probably the ideal way, and the way that we use this in the lab, is we do both a varying allele we do both a mouse cell depletion followed by a hybrid genome alignment, and that really makes sure that you don't have any, if there's any residual nucleic acids, they don't align spuriously. So the data that I've shown so far has been with a targeted sequencing panel, but we've used this on much larger genomic territory. Here's a study that we did using whole exome sequencing on paired PDX, and I'm showing just the most common mutations in panel A in the top left. We were able to do some other interesting things with this as well, including looking at mutational signatures, which are shown in panel B, comparing mutations called between samples before and after chemotherapy. And in panel D, we were able to get reliable copy number calls using this approach. And what we really wanted to use this for was to be able to build a database of highly annotated patient-derived xenografts, and our goal was to be able to mimic the great cohorts of human primary samples that are now available. And so we've been using the CBio portal as kind of a warehouse for our data, and we're trying to build a very well annotated public database of xenografts. The type of data that we can get from this shows both clinical events, which can include surgery, treatments, treatment timelines for the patients, as well as a genomic overview that includes copy number changes and mutation calls. Everything that I've shown so far has been data that we've published. I also want to show a couple of slides of unpublished data, techniques that we're starting to do in the lab using this approach. One is we're very interested in the idea of in vivo genome engineering of patient-derived xenografts, and this really depends on being able to get reliable infection of lentiviruses into cancer cells. And so we begin with making a single-cell suspension from a patient-derived xenograft. We then remove stroma, and that's depicted here in the middle. The human fraction initially is actually less than 25% in this particular model, but after we deplete the mouse cells actually end up being the minority cell population. So this enriches for cells that we actually want to infect with lentiviruses. We do an ex vivo spin infection, and then as a proof of concept, we infected this suspension with a lentivirus that expresses mCherry. I think you can appreciate that you can get a significant fraction of positive cells after this. You can even image this using optical imaging techniques. So this was kind of our proof of concept pilot experiment, that we could do this, and we started to think about other approaches that or what we might be able to use this technology for. So one approach has been, we've been thinking about how we wanted to do, or how we'd like to use this for target validation. So we developed these constructs that are similar to other constructs that have been used by other groups that are lentiviral vectors that express either cypridina luciferase or Gaussian luciferase tied to the expression of a shRNA of interest. And what this allows you to do is look at different luciferase expression as a surrogate for that shRNA. And this allows you to do clonal competition experiments to compare the fitness of the gain or loss of different genes. And so the idea here is basically at time 0, you do a spin infection with lentivirus, engraft your tumors, wait until the tumors start to become established, and then you add doxycycline, which turns on the shRNA and turns on the luciferase. And then what we do is just measure the luciferase signal in these mice over time, which is shown here on the right, and the gap in luciferase signal that develops over time is really an indication of the fitness of the gain or of the loss of a specific gene. So in this example, this is just showing a negative control shRNA targeting Ranilla versus a positive control that targets RPA. And we've also been thinking about doing more exotic things. So this is a similar vector where instead of using luciferase, we're now bringing in the programmable nuclease Cas9. And we wanted to be able to select cells that actually express Cas9. And we thought one nice approach to be able to do this would be to use immunomagnetic sorting. And so we created a new type of reporter that's a CD4 protein that is truncated to have only domain 1 and 2. And what this allows us to do is use super paramagnetic beads to pull out the cells that express this truncated CD4. At the bottom here, I'm just showing a schematic of how this experiment would work. Basically, initially, we start with a PDX. We infect with a lentivirus shown in blue, and then after the next passage, we do an immunomagnetic sorting. And then in the absence of docs, you don't get expression of Cas9, but in the presence of docs, you get expression. So this allows us to have inducible genome editing in PDX. And we've done some pilot experiments to see just how much purity we can get from a immunomagnetic sorting approach such as this. And the x-axis here, we basically spike in a percentage of cells that are positive for CD4 from the start. And on the y-axis, I'm showing the percentage of cells that are positive for CD4 after immunomagnetic enrichment. So the black line is really just showing that when we start with 20% or 10% CD4 positive cells, after one enrichment, we can be higher than 60% or higher than 80%. So this was just to prove to ourselves that we could bring these cells, purify these cells out. I also just want to show a couple of slides of unpublished data for some of the single-cell RNA-seq that we've been doing. If you want to be able to do single-cell RNA-seq, it helps to be able to get your cells into a nice single-cell suspension. So we've been collaborating. This is data from Ashley Lofney from Joan Massaguet's lab, and the sequencing and data analysis is done in collaboration with Donna Pear. And what this is showing is a collection of more than 40,000 cells from a number of patients that we've been sequencing, and these are clustered using a typical algorithm that's often used for single-cell RNA-seq. This is sometimes called a t-SNE plot. And what this is just showing is that the cells that we're sequencing are not clustering specifically by one patient. These are colored by individual patients. And this is telling us that we're not getting a lot of patient-to-patient variation, or that is that the patient-to-patient variation is not really driving the variance here. And so now this is the same data, but it's now, instead of being colored by patient, it's being colored by cell type. And so we're pulling all different kinds of cell types out of this. Many of these are infiltrating immune cells, but many of these other cell types are the cancer cells themselves. The way these break down is the majority of cells that we're sequencing here are the lymphoid cells in blue and myeloid cells in this kind of magenta color. I think if we did more of the sequencing, we would probably think about depleting a fraction of these immune-infiltrating cells so that we can sequence a higher fraction of the epithelial cells. But this is just to point out that we're able to really characterize these individual cell populations. And I mentioned that one of our key factors here is we wanted to make sure that from patient to patient, the variability that we're studying is the biological variability and not differences, technical differences from sequencing run to sequencing run. And so what I'm showing here is, again, the patient ID coloring on the left, which is similar to, which is basically just showing, again, the data from an earlier slide, and then colored by the Shannon Diversity Index, which gives us a sense for how different each group is, or how different each single cell is from its neighbors. And so you can see that all of the cells that were previously colored as lymphoid cells are all mixing well together, whereas the components of this that are from the epithelial cell compartment are the ones with higher diversity. The interesting thing about this data, if you look at the violin plot at the bottom, is that there are two samples that have really very low diversity. This one shown in magenta, and then this other dark red sample. And these correspond to these patients up here. And the interesting thing is that these samples both come from metastatic samples. So this is telling us that there's some metastatic, there's some bottlenecking that's happening that's coincident with the metastatic process. So to summarize some of the, what I've just told you is that we're using automated dissociation to improve the consistency within treatment groups for PDX studies. It allows for us, for downstream sorting applications that include flow cytometry and immunomagnetic enrichment or depletion. It can be used to generate cell suspensions that are suitable for virus infection. And we also think, in our hands, it's an ideal partner for single-cell sequencing. So with that, I'd be very happy to take questions for people who are interested about these topics and others related to patient-derived xenografts.
Excellent presentation, Dr. Poirier. Thanks for bringing that information to us. Before we get started with all of your questions, here's a quick reminder about how to reach us today. Questions can be sent via the green Q&A button at the lower left of the presentation window, and we'll get to as many as we can. Our first question is, would the use of flow cytometry in conjunction with single-cell dissociation make primary tumor identification easier?
I think it has the potential. I think it would depend on having good markers for immunophenotyping for the particular type of primary sample that you're trying to identify. I think one thing you lose when you dissociate tumors is that you lose all the morphological information that you would have on a tumor that's been sectioned. So in some respects, I think it could be useful. In other respects, you lose some information. Here, from a clinical perspective, we're working with a minority of tumors where we have fresh surgical or biopsy samples available. I think the majority of cases, you're working from FFPE, and I think that this technology wouldn't be applicable in that case. But in other cases where you have fresh tissue and good markers, I think there's potential there.
Does the presence of mouse stromas in the human cells and tissues enhance the tumor take rate?
That's a really good question, and it's not something that we've looked at systematically yet. I can tell you that anecdotally, when we've purified just the human cancer cells away by fax and then injected those into a mouse, they take very well. In the handful of samples that we've looked at, and that sort of mirrors the experience that we have with injecting cultured cell lines directly. That said, we know that some PDX types take better than others, and some, for example, different breast PDX require addition of estradiol and things of that nature to help improve the take rate. So there are certain cell extrinsic factors that do matter. I think one of a very significant challenge in the future for PDX is going to be trying to replicate a stromal environment that's as close as possible to the human environment in a mouse. For example, we know that different growth factors don't have activity, cross-reactivity between human and mouse, and so those factors and others probably play a role in the differential take rate that we see between different tumor types that seem to be independent of proliferation rate. So I hope that's a useful answer, but I can't really give you a definitive answer based on experimental evidence from my group.
Will this be available to track children as changes happen due to infections, environment, etc., to map the genetic variants through time?
I think in this case, by children, you mean ancestrally for the tumor, and if that's the case, we have tracked over multiple passages. For example, the amount of tumor stroma that's there. We have done sequencing after multiple passages to look at copy number and single nucleotide variants. If your question was more related to clonal lineage tracing with lentiviruses, my group hasn't done that, but it's something we're interested in doing, and there are libraries that are publicly available, for example, from AdGene to barcode and lineage trace using integrating lentiviruses.
Have you compared orthotopic to subcutaneous PDX, and do you think clonal parity with the original patient tumor is influenced by orthotopic versus subcutaneous mouse stroma?
This is another great question. We haven't looked at this systematically in my group. I know that there are some groups, particularly in cellular immunology, I can't quote the literature to you, but that have looked at the immune cell infiltrates specifically in primary tumor or in subcutaneous flank xenografts versus orthotopic versus primary tumors, and there are significant differences there in the types of immune cells that infiltrate and their phenotype. We haven't looked specifically at the clonal heterogeneity of xenografts at different sites, but we do know that in metastatic sites you do get clonal bottlenecking, and we see very significant differences in response rate to different types of therapies depending on the anatomical context. So I think it's an important question. I think it's a challenging one that my group hasn't specifically addressed yet, but I think it should be done.
Does clonal heterogeneity change over time?
There are two great papers that I know about this subject. One of them is a little bit older. It comes from, it's in a Nature letter from Sam Apariccio's group at the University of British Columbia. They look specifically at breast cancer patient xenografts at single-cell resolution, and they were able to see a few different modes of clonal evolution happening. Some PDX had relatively stable clonal proportions, which suggested that maybe there wasn't much selection going on, while other PDX saw significant selection of specific clones that was reproducible in multiple replicates. That, I think, is a kind of a hallmark paper in the literature. There was another paper in Nature Genetics, the most recent issue from Todd Gollop's group. They looked at tumor evolution, specifically looking at copy number alterations in more than 1,000 different PDX from 24 cancer types. So certainly that's one of the more broad examples of looking at clonal evolution in these models. And I think that their general conclusion was that there is a mouse-specific tumor evolution process that's happening. I would encourage you to read through that paper.
What system did you use for single-cell sequencing?
We have a few different systems and approaches that we use, but the data that I showed here where we're sequencing off of fresh cells is with the 10x genomic system.
Is it possible to quantitate changes in gene expression within the stromal cell component?
Sure. So the data that I showed you here, where we aligned to the hybrid genome, we were really just interested in what was happening in the human cells, so we just discard the reads that preferentially map to the mouse genome. It would be perfectly reasonable to count those reads and assign them specifically to the mouse stroma.
Are there special technical requirements or challenges for aligning reads to hybrid genomes?
I think this was something that was more challenging to do a few years ago than it is now. The major requirement is that for most aligners, the human reference and the mouse reference have to both fit in memory at the same time. And that means that the program that's doing the aligning has to be able to address a chunk of memory that large. And for example, BWA, which is the aligner that we used here, it used to be only available in a 32-bit application that was unable to address the amount of memory required to align against such a large reference. Now there are binaries available for that. Most operating systems and hardware support 64-bit, but it's something that you should be aware of and pay close attention to with respect to the memory requirements.
How do you screen for cross-contamination of PDX samples and avoid them?
This is a really critical issue, and I think that there have been a few papers kind of criticizing the literature and the scientific community for not paying enough attention to this. I think it's a major problem. One thing, our approach used to be to take a similar approach to cell line authentication using short tandem repeat to try and get the identity of different PDX. I think the problem with that is it's fairly expensive. I think commercially doing that, it's more than $100 per sample, and we weren't able to do that internally. More recently, what we've been doing to check for major contamination is to use SNP qPCR assays in dry down plates and PCR to genotype between 8 and 16 SNPs by direct lysis from frozen vials. So basically you can scrape the vial, do a direct lysis to get the DNA out, and then do a few different qPCR assays to get the genotype. This works for catching this works for catching kind of major contamination, but it's not very sensitive. So I think one thing we also pay very close attention to is, especially if we're concerned with minor contamination, is very high-depth sequencing of informative variable SNPs. So in the MSK impact assay that I told you about, we capture about 1,000 specific SNPs, and we compare the genotypes of those SNPs to the normal blood from that patient, but also to previous passages. You can also estimate whether there's minor contamination based on those SNPs in terms of the number of those SNPs that have that are either homozygous, heterozygous, or triallelic, and so we pay close attention to that. I think that probably in the future people should start publishing these SNP fingerprints at least for 8 to 16 SNPs for PDX that they use in publications.
Let's see. The next person says, "Hi, Dr. Poirier. It was an excellent talk. I work on PDX samples, and I grow stem cells in the form of spheroids from the cells isolated from PDX samples. I would like to know, how can we harvest these stem cells, spheroids, for RNA or protein by effectively removing dead cells?" And then there's another part of it too, but we'll ask that first.
That's a tough question. So, my lab works with small cell lung cancer, and cell lines that are generated from small cell lung cancer tend to grow in these spheroids, and we recognize as well that you have a lot of dead cells at the center of these spheroids that don't get exposure to nutrients, or they don't get enough oxygen. And maybe the population that you're most interested in is the cells that are really at the interface with the liquid medium. I can't give you a definitive answer, but I think that one approach could be to use flow sorting with a live dead dye, and then sort the live cells directly into Trizol for RNA-seq. That could be one approach. I don't know whether that would introduce additional biases, but that's an approach that we've used effectively for getting RNA-seq data from isolated immune cell populations, for example, from flow cytometry.
Okay, and here's the next part of their question. Centrifugation at 49 grams, I believe it's grams 49G, or histopack hasn't been very effective. Your suggestion will be very valuable. Thanks.
I think I would stick with flow there, unless you have unless the fraction of cells that you have is very, very high in terms of the percentage of dead cells. Then you might consider maybe a two-part application where you could try to remove dead cells by, phycol, like you suggested, or maybe perhaps by an immunomagnetic dead cell removal kit followed by FACS. I think that would certainly be the way to get, pure single cells directly into RNA-seq. For RNA-seq could be to sort them directly into Trizol.
Okay. And I just want to say that, here we go, another one. So if serially passaging these primary tumors, presumably the mouse stroma is not metastatic and would die when subsequently injected in following passages. I guess it's a comment for you to comment on.
I mean, yes, we believe that to be the case. So we've done experiments where we infect a single-cell suspension that contains human and mouse cells with a lentivirus, and when we passage that tumor and dissociate a second time, the only cells that are marked with the lentivirus are the human cells. So we interpret that, of course there are other interpretations, but we interpret that to mean that, because of the Hayflick limit and the points that you've mentioned, that probably those cells are not sticking around for many passages.
Many questions have come in, so if we didn't get to yours, we will follow up with you via email. I would like to once again thank Dr. J.T. Poirier for his presentation, and I would like to thank our sponsor, Miltenyi Biotech, for making it possible to bring this presentation to you. Today's webcast will be available for on-demand viewing through April 2018. You'll receive an email from us alerting you when it's available on demand and posted on labroots.com. You're welcome to forward this announcement to any colleagues who weren't able to join in today. Thanks for logging on and participating in today's broadcast. We will see you next time.